Welcome to the Passionate Minds course content review. Today we'll be covering cell structures and we'll have a focus on specific eukaryotic cell structures. You'll also review how these structures help in the functioning of the cell. So let's first talk about the plasma membrane. So the plasma membrane is found in all types of cells, such as prokaryotes, eukaryotes, plants, and animal cells. So the plasma membrane is a very thin barrier that consists of a lipid bilayer. So a lipid bilayer, like we discussed in the lipid lecture, and it consists of proteins as well that we will see in the fluid mosaic model, and that will separate the exterior environment from the interior environment. So we see that there's two very different environments. So the plasma membrane consists of a semi-permeable barrier, which allows for the passage of small molecules like oxygen, nutrients, and waste to support the cell volume in general. And it also has a very key role in cell communication, cell adhesion, and cellular structure. So the cell part is made up of a lipid bilayer, like I said over here, and specific associated proteins. And a dysfunction results in cystic fibrosis. So the endomembrane system is a continuous sort of system, and it's connected indirectly via vesicles, and it plays a big role in the cell's compartmental organization. Right? So the endomembrane system, the term endo means within. So it is a group of membranes and organelles in the eukaryotic cells that works together to modify, package, and transport lipids and proteins. It also includes a variety of organelles, such as the nuclear envelope, lysosomes, and vesicles, endoplasmic reticula, and the Golgi apparatus, which we'll cover shortly in the next slides. Technically, it's not a part of the cell, but the plasma membrane is part of the endomembrane system. So we'll see that the plasma membrane interacts with other endomembrane organelles, and it's the site usually where specific things like proteins are secreted. So, for example, in the pancreas, you have enzymes that are secreted. Note that the endomembrane system, however, does not include the mitochondria, chloroplasts, and peroxisomes, so these aren't involved. So the nucleus is found in all eukaryotes, so not prokaryotes, but eukaryotes, which includes animal cells and plant cells in particular. So the cell part is found within the cytoplasm, and it's the most visible organelle. So when you see under a microscope, usually that's the most visible structure that you'll see within the cell. So the cell part, it contains most of the cell's genetic material, and you'll see that the DNA, it is organized into chromosomes. It's tightly wound up, and then it's organized into chromosomes, which play a role in storing the genetic information. The cell part consists of a double membrane nuclear envelope. So I'll write that here, nuclear envelope, and that will enclose the nucleus. So you have these nuclear pores, like we see over here. These nuclear pores will regulate the entry and exit of molecules from the nucleus. And finally, you have a nuclear lamina, which is present, and the nuclear lamina is what helps regulate the nucleus's shape. So it's a protein matrix that will help regulate the shape of the nucleus. A dysfunction leads to hereditary diseases just because it's genetics related. So like Down syndrome, for example. So trisomy 21. The nucleolus is found in eukaryotes. It is found inside the nucleus. That's key. It's found inside the nucleus, but it is not membrane bound. Okay, so it's not membrane bound. Basically, what it is it's a sort of visible density that's within the nucleus, and the appearance of the nucleolus differs across the cell cycle. So at various stages, it looks different. So the main role of the nucleolus is that it is the site of rRNA synthesis and ribosome assembly. So the cell part is made up of rRNA genes, rRNA as well, and the associated proteins present with it. So a dysfunction of the nucleolus leads to programmed cell death, which is essentially cancer. The rough endoplasmic reticulum is found in all kinds of eukaryotic cells. It is found in, specifically it's found in the cytoplasm as a membranous structure. And the purpose of the rough endoplasmic reticulum is that it synthesizes proteins and glycoproteins. So it synthesizes proteins and glycoproteins. So glycoproteins are basically proteins that are modified with covalently linked carbohydrates. So you think of a protein like this, and then let's just say you have these carbohydrates that link from it. So what is synthesized is what proteins are synthesized include the proteins of the endomembrane system, proteins that are embedded to the outer cell membrane, and proteins that are secreted from the cell. So these include protein hormones and enzymes. Additionally, it can produce transport vesicles, which distributes lipids and proteins to other components of the endomembrane cell. So it can produce transport vesicles, which can be used for other components of the endomembrane cell and endomembrane system, or eventually end up at the plasma membrane. So this is why the rough endoplasmic reticulum is given the name membrane factory. So the cell part is made up of it's just continuous with the nuclear envelope. It's a membranous structure, and combined with the smooth endoplasmic reticulum, it accounts for half the total membranes in many eukaryotic cells. It also has an internal component called the ER lumen, and the surface of the rough endoplasmic reticulum is studded with ribosomes. So that's one of the key features that we see over here. The surface of the rough endoplasmic reticulum is studded with ribosomes. A dysfunction leads to improper protein folding, because a rough endoplasmic reticulum is responsible for a lot of proteins synthesis and production, as we talked about in terms of its purpose. The smooth endoplasmic reticulum is also found in all kinds of eukaryotic cells. It is a membranous structure found within the cytoplasm, similarly to the rough endoplasmic reticulum, but the difference is that it is smooth, meaning that there's no ribosomes that are embedded onto the surface. So the smooth endoplasmic reticulum has four main roles. So we refer to it generally as a biosynthetic factory of the cell. So what it does is it can synthesize lipids, it metabolizes carbohydrates, it detoxifies drugs and proteins, and it stores calcium ions. Additionally, the smooth endoplasmic reticulum doubles in size when detoxifying, but after a waste has been processed, it returns to a normal size. So the cell part is, once again, it is continuous with the nuclear envelope, continuous with the rough endoplasmic reticulum as well, and it also accounts for half of the total membranes in many eukaryotic cells. Combined with the rough endoplasmic reticulum, it accounts for half of the total membranes. The internal compartment, once again, is also known as the ER lumen, but the surface is smooth and it's lacking ribosomes, just like I've mentioned. A dysfunction results in Huntington's disease. Ribosomes are found in all types of cells, so this includes prokaryotes, eukaryotes, plants, and animal cells. So the cell part is found either in the cytosol as free cytosolic ribosomes, so this is in all cells. So all cells, all cell types in particular, consist of ribosomes that are free in the cytosol, so free cytosolic ribosomes. But in eukaryotes specifically, what happens is they're found in the surface of the rough endoplasmic reticulum. So this only occurs in eukaryotes. It is also found in mitochondria and chloroplasts, which are also found in, which are specific to the eukaryotes. So the main goal of ribosomes is to carry out protein synthesis through the translation. So there's a process called translation, which we'll talk about in molecular genetics. So translation of RNA into protein. So the cell consists of small complexes, both made up of rRNA plus proteins, and it is not membrane bound. That's a really important detail. Not membrane bound. Okay, so we see that there's two complexes over here. You have a small subunit and you have a large subunit. Additionally, a dysfunction in the ribosome leads to something called dyskeratosis congenita. So the Golgi apparatus is found only in eukaryotic cells. It is in close proximity to the endoplasmic reticulum, but it's basically found in the cytoplasm, just like every other part that we're looking at. The main role of the Golgi apparatus is that it is involved in shipping and receiving. So it's a shipping and receiving center of the cell, and it is able to modify products that come from the ER. So it modifies products from the endoplasmic reticulum. A main modification is called glycosylation. So glycosylation, and this is basically when you add carbohydrate groups to proteins and lipids. Another function of the Golgi apparatus is that it manufactures some macromolecules, such as carbohydrates. So what it'll do is that it can sort and package material, these materials like carbohydrates into transport vesicles. Thirdly, there's also another important distinction that we must consider here. You have a cis phase and a trans phase. So the cis phase is where the receiving side of the Golgi apparatus, and the trans phase is the shipping side of the Golgi apparatus. So that is where vesicles will bud out. So let's go through a little process here. So the transport vesicles from the endoplasmic reticulum will first fuse with the cis phase, and then they'll empty their contents into the lumen of the Golgi apparatus. So you see that the lumens are these little spaces between here. So then what will happen is that proteins and lipids, they'll travel through the Golgi and they're modified, and then they'll be packed and sorted accordingly, and then finally vesicles will bud out from the trans face of the Golgi apparatus, and they'll be sent to their destination. The Golgi apparatus basically consists of flattened membranous sacs that are called cisternae, like we see over here. So these look like sort of pancakes in a way. Finally, a dysfunction in the Golgi apparatus leads to achondrogenesis. The lysosome is found in all eukaryotes. We're currently questioning whether it is found in plant cells, but it is. We'll currently say that it is found in all eukaryotes. The cell part is basically what it is it's a membrane-bound compartment that is found in the cytoplasm of the cell. Now there's three main functions here as to what the cell part does. We'll go through each one. So the first function is enzymatic hydrolysis of various macromolecules. So these macromolecules include lipids, fats, proteins, and nucleic acids. So it performs digestion using enzymes. So the lysosome contains specific enzymes that, if it will fuse, it'll break down components. So it'll fuse with food vacuoles, for example, and it will absorb all the contents, and then it'll digest it, breaking it down to part that could be useful for the cell. Lysosomes are also able to engulf another cell by phagocytosis. By forming something called a food vacuole. So a lysosome will fuse with the food vacuole and digest the molecules inside of it. So that's how some cells can engulf another cell. Finally, lysosomes can also recycle the cell's own organelles and macromolecules in a process that is called autophagy. So autophagy refers to self-eating. The cell part is made up of a lysosomal membrane. So there's specific properties to the lysosomal membrane which allows the lysosomal compartment to be more acidic than the rest of the cell. So this is like an enhanced environment so that you can have more variety of enzymes that are involved here. The size, number, and shape of the lysosome are often variable though. Dysfunction in the lysosome can lead to something called eye cell disease. Vacuoles are found in all eukaryotes, so plants and animal cells. Vacuoles are a membrane-bound structure that is found within the cytoplasm, and it is derived from the endoplasmic reticulum and Golgi through the endomembrane system. So that's how you form a vacuole. So it is a result of the endomembrane system and the interactions within the previous organelles that we've talked about. It is predominantly for storage, but it varies by the cell types. So food vacuoles are formed by phagocytosis. So phagocytosis produces food vacuoles. You have contractile vacuoles, which is done by protists. So protists have contractile vacuoles to pump out excess water from their cells. And finally, you have so plants, they have a central vacuole. So central vacuole helps to hold organic compounds and water. So in plants in particular, they have one large central vacuole, but in animal cells, there's many smaller vacuoles that are present. So we see this example over here. We have the plant cell. There's this one large central vacuole here. Additionally, if you have a dysfunction in the vacuole, you're going to have something that's called Danin's disease. Peroxisomes are found in all eukaryotic cells, and they're found within the cytoplasm, just like all the other cell parts. So the cell part consists of having specialized metabolic compartments to break down organic molecules by oxidation. So it breaks down organic molecules by oxidation. And as a result of this oxidation, you're going to produce hydrogen peroxide. So hydrogen peroxide is unstable, so what will happen is that it is converted to water and oxygen then. Peroxisomes also perform reactions with different metabolic functions. So they can break down fatty acids. So it breaks down fatty acids and amino acids necessary to produce other biomolecules such as cholesterols and phospholipids. So these can be reused. The cell part is made up of a single so it's basically a single membrane with a single crystalloid enzyme core. So this is called a crystalline core. It has an enzyme function. The mitochondria is found in all eukaryotes, so this includes plant and animal cells. It is found within the cytoplasm, and it carries out a function that is called oxidative metabolism. What does that mean? It means that it is a metabolic process that converts energy from macromolecules to ATP using oxygen. So it will ultimately produce ATP using oxygen. The cell part consists of a double membrane. So we see one membrane here, and then we see a second membrane over here. And the outer membrane is very smooth, but the inner membrane, it folds into these little folds here that are called cristae. So the reason why they're folded into cristae is to increase its surface area. The inner membrane will create these two aqueous compartments. So you have the outer compartment, which is the intermembrane space, and then you have the mitochondrial matrix, which is the internal compartment over here. A dysfunction in the mitochondria leads to congenital lactic acidosis. Chloroplasts are found in plants and algae, and they're found within the cytoplasm. The main function of chloroplasts is to carry out photosynthesis. So photosynthesis is a process which uses sunlight to fix carbon dioxide. So to fix carbon from carbon dioxide to generate organic molecules that are energetically rich, such as glucose. So the chloroplasts are surrounded by two membranes. So you have an internal compartment, like we see in this diagram here. There's an internal compartment that is filled with an aqueous solution, and this aqueous solution is called stroma. So stroma. The internal thylakoid membrane. So we see these thylakoids over here. So the internal thylakoid membranes, that's what contains chlorophyll, and they form these membranous sacs. You call each one of these discs that are stacked, they're called grana, and inside the thylakoid is the thylakoid lumen. A dysfunction results in leaf chlorosis. Now let's talk about the structural components, so the structural parts of the cell. The cytoskeleton is found in all types of cells. So prokaryotes and eukaryotes, they all have a cytoskeleton. The cytoskeleton is found as protein fibers. So protein fibers that are extended through the cytoplasm, like we see in this diagram over here. So the cytoskeleton has many functions. It's able to organize the cell's structures and activities by anchoring many organelles. So it organizes by anchoring organelles. It helps to support the cell and maintain its shape. So maintains cell shape. It is also able to interact with motor proteins to enable motility. And finally, the cytoskeleton enables vesicles to travel along its tracks. So the cell part is made up of actin filaments, microtubules, and intermediate filaments, and a dysfunction leads to Alzheimer's. So before we talk about centrosomes, let's first go through a little bit of vocabulary here. So this is a centriole and this is a centriole. A centrosome consists of a pair of centrioles at right angles to one another. So you can see how this forms a right angle because it's perpendicular. So when you have two centrioles in this arrangement, you form a centrosome. So let's answer the first question. So a centriole is found only in animal cells. In plant cells, they have their own version where they have microtubules that originate from a small nucleation site. The cell part is found near the nucleus, and microtubules are able to grow out from the centrosomes. So it looks something like this. So you see in a cell diagram, you see microtubules that are extending, they're projecting out. So there's two main functions over here. So the first function is that the microtubule portion of the cytoskeleton is involved in chromosome separation during cell division. So in anaphase, what happens is that you have the cell over here, and then we have one centrosome here, we have another centrosome here, we have the mitotic spindle fibers over here. Here, let's just say this is a chromosome, these are chromosomes. So in anaphase, you have them pulling towards the poles of the cell, and that is a result of the centrosome working in action. Secondly, the microtubules from the animal cell are involved as microtubule organizing centers. So like I said before, microtubules grow out from a centrosome near the nucleus in animal cells, and a dysfunction leads to cancer. Cilia and flagella are found in prokaryotes mostly, but they're also found in some eukaryotic cells like sperm. The cell part, basically what it is that it has microtubules that control the beating of the cilia and flagella. So they're locomotor appendages. That's the term to describe them, locomotor appendages. Basically, they're these extensions of the cell. The cell part, what it does is it basically acts in propulsion. Propulsion key for movement of the cell. It basically works to move around various locations from point A to point B. The cell part is made up of microtubules, and then I'll talk about the specific associated proteins. So cilia and flagella, they have a common structure, so they have a core of microtubules that are encased by plasma membrane, and then they have this basal body that anchors the so you have a basal body that will anchor the cilia and flagella, and then you have a protein that is called dynin. So this is a motor protein that drives the bending movements. Finally, what differs flagella from cilia is a protein that is called flagellin. Flagellin is what enables the flagella to move in a rotary pattern. So it's able to move in the circular fashion. So that is what differentiates the prokaryotic flagella from cilia. A dysfunction leads to male factor infertility. So cell walls are found in all types of cells. So prokaryotes, eukaryotes, plants, and animal cells. And this is what distinguishes plant cells from animal cells. Prokaryotes and fungi, they also have cell walls too. So the cell part, basically what it is a very, very thin barrier that consists of a lipid bilayer and proteins that separates the exterior environment from the interior environment, just like I talked about for the plasma membrane. But in this case, I'll be more specific to plants because this is what distinguishes plant cells from animal cells in particular. So the function of a cell wall is to protect, maintain the shape, and prevent the excessive uptake of water in plant cells. So protect maintains the shape, and it will prevent the excessive uptake of water. So prevents the excessive uptake of water in plant cells particularly. Okay, so the cell part is made up of peptidoglycan, if it is prokaryotic. So peptidoglycan is a polymer of sugar and amino acids, and this is notably found in only prokaryotic cells. Plant cell walls are made up of cellulose, and cellulose which is embedded in other polysaccharides and proteins. So this is specific to plants. A dysfunction leads to bacterial soft rot.